I, as I said, I'm going to present my study entitled Study of Cyprideis remanei valve morphology as an indicator of paleoenvironmental change in Lake Nyalonzwele in Inhambane, Mozambique. Paleoreconstruction has been used to explore the impact of environmental change on human society, such as samplement patterns and food resource availability. To study these effects, ostracods bivalve, crustacean found in freshwater and marine environments, has been used. And among them, Cyprideis, a genus known for its high tolerance to salinity variation, was selected due to extensive study in paleoenvironmental research. Data was collected from Lake Nyalonzwele in Inhambane province, which contain a rich fossil record of Cyprideis, offering insight into environmental change and their influence on humans. So these lead to the question, how can valve morphology serve as an indicator of paleoenvironmental change during the middle and the later Holocene? 80 different samples with a total of 327 left valves from adult individuals were analyzed. This selection of 8 samples analyzed was based on section that a server were on the lacustre environment allowed for continual preservation in relation to the section that a server where the number of ostracod preserved varied. So photographic image of the valve were captured using a binocular microscope, and morphological data were then acquired by collecting 2D coordinate of anatomical landmarks and semi-landmarks valve, employing geometric morphometric techniques that enable to precise identification of shape variation. The cruscal walls test on centroid size within the female subgroups reveal size difference among the samples, notably of sample 7 and sample 3, showing the distinct median position compared to the others. In the PCE2 reveals overlapping morphospace among the samples, but highlighting significant difference between a few. For example, in PCE2, environmental factors has salinity drive morphological variation has seen in the grid transformation. Among the 2 samples characterized by period of environmental stress and an increase in valve size and shape, we select the sample 3 as an example for our finds. This sample displaying a larger median valve in terms of size compared to the more recent sample, and showed a transition from an arid to a humid climate. And also paleoenvironmental data such as sea level retreat and drier condition align with a reduced forest cover in regions like KwaZulu-Natal, and similar climatic stress likely impacted Mozambique, has reflected in morphological valve, indicating a regional response to changing environmental conditions. The morphological change observed in Cyprideis remanei valve in this study suggests an environmental shift in Mozambique in period between 2006 to 2001 BP. In this way, the transition to a drier climate led to the migration of hunter-gatherers and a change in diet toward a lifestyle more associated with the exploration of coastal resources. The convincing hypothesis is that Mozambique has any corridor for the Bantu groups when there was greater migration during this period. And the archaeological records indicate a shift from hunter-gatherers to Bantu speaking groups at the archaeological site of Matola and Eduardo Mondlane University camps in Maputo around 1050. Evidence for the presence of ceramic remains iron slag agriculture practice suggests that these groups were already established and practicing trade routes in the Indian Ocean. The ecological records of Cyprideis analyzed through the developed technique offer versatility application, such as identifying patterns in the archaeological records and highlighting period of stress potentially linked to the cultural and economic shift under the climatic conditions. And I want to say thank you for your attention and thank you for the following institution and project to their contribution to the realization of my work. And here I have some reference that I use to do my work. Thank you.